What if I told you we had a tool so powerful it could literally rewrite the genetic code of an entire species? A technology that could, potentially, wipe out ancient diseases like malaria for good, but that also comes with risks so massive they could change our ecosystems forever? Well, let's get into it. We're talking about something called gene drive.
So to understand why scientists are even thinking about something this radical, let's start with a basic question. What do you think is the deadliest animal on Earth? Seriously, what pops into your head? A shark, right? Maybe a lion? Or a tiger?
Well, the answer is actually way smaller. It's this, the mosquito. By a massive margin, this tiny insect is the single most dangerous animal to human beings on the entire planet.
And that gets us to the true scale of the problem here. I mean, this isn't just about some annoying, itchy bites. No, mosquitoes are carriers for some of the most awful diseases we've ever faced. We're talking about malaria, which puts half the world's population at risk. Dengue fever, which is just spreading like wildfire. Zika, with its absolutely heartbreaking consequences. It adds up to hundreds of millions of people getting sick every single year.
And as this report from the Takshashila Institution points out, the toll is just immense. For places like India, these aren't just a health crisis. They're an economic disaster, crippling communities, overwhelming healthcare systems. And that is why we're looking for a solution as game-changing as a gene drive.
So how in the world do you fight back against a predator that relentless? Well, scientists are now working on a technology that doesn't just swat a few mosquitoes. It actually aims to rewrite the DNA of the entire species.
It's called a gene drive. Now, at its heart, it's using CRISPR. You've probably heard of it. That amazing tool that works like molecular scissors for DNA. But here's the wild part. A gene drive is designed to cheat genetics. Normally, a gene has a 50-50 shot of being passed on. A gene drive, though, it makes sure it gets inherited almost 100% of the time, spreading like crazy through a population.
This is a great way to think about it. Imagine you're in a document and you use find and replace. But imagine that every time you replaced a word, the instructions for find and replace got copied right alongside it. So the next generation of that document could automatically do it again and again and again. That's a gene drive. It's a self-perpetuating genetic chain reaction.
Okay, so you've got this incredibly powerful tool. Now what? Well, scientists have a huge choice to make, and it leads down two totally different paths. Do you use this power to try and erase the entire species, or just edit it?
Let's break these two strategies down. On one side, you've got population suppression. This is pretty much the erase button. The idea is to engineer a trait, like making all the females sterile, that spreads until the entire mosquito population just crashes and disappears from an area. Then you have population replacement. This is the edit option. The mosquitoes get to live, but we tweak their genes so they just can't carry things like malaria or dengue anymore.
But here's the catch with trying to erase them. It's not a one-and-done deal. You have to keep releasing new modified mosquitoes year after year. Because as you kill off the population, your gene drive dies with it. And all that pressure gives the few survivors a massive incentive to evolve resistance and come roaring back.
And the catch with the edit option, well, in some ways it's even scarier. Because the change you make, it's permanent. It stays in the ecosystem forever. There's no undo button. And that right there is the huge dilemma. Once you let a self-copying genetic change like this out into the world, you can't really call it back.
So that brings us right to the heart of this whole debate. When you have a technology this powerful and this permanent, you have to ask, what could possibly go wrong? The answer, it turns out, is a lot.
We can sort of break these risks down into three big stages. First, when the tech is created in the lab. Second, when it's deployed out into the real world. And finally, the long-term monitoring, seeing what happens to the ecosystem. Let's start at the beginning, in the lab.
You know, the risks don't just start when these mosquitoes are flying free. They begin way before that, inside these high-security labs where scientists are building them and government regulators are supposed to be watching over the whole process.
So even in this super-controlled environment, things can get messy. That CRISPR tool, it might snip the wrong piece of DNA, causing weird, unintended mutations. The mosquitoes themselves could start evolving resistance to the gene drive right there in their little lab gauges. And of course, there's always the nightmare scenario, an accidental escape.
But as tricky as the lab part is, the moment you open that door and let these organisms out into the wild, the complexity just explodes. Let's move on to that deploy stage.
So for a gene drive to even work, you have to release it in the right place.
But what if your map is totally wrong? Check out this statistic from the report. Official numbers on dengue in India are thought to be underreported by nearly 300 times. 300! Imagine trying to deploy this tech when your data is that incomplete. You're basically flying blind.
And that's just one problem. Mosquitoes, they don't care about our maps. They fly around, migrating from other areas, and that can water down the whole effect. But maybe the scariest risk is vector switching. Imagine you spend a decade and millions of dollars wiping out the main malaria mosquito, only to find out the malaria parasite just moved house and started infecting a completely different species. The whole effort would be for nothing.
And that leads us to the final stage, and honestly the biggest unknowns of them all. Once that gene drive is out there, spreading year after year, decade after decade, what does it do to the rest of the ecosystem? This is the monitoring phase.
Here, you're talking about huge ripple effects. If you get rid of a mosquito species, what happens to all the fish and birds and frogs that eat them? There's a small but terrifying chance the gene drive could somehow jump to a totally different species. And maybe the most likely problem, nature hates a vacuum. If you eliminate one species, you create an opening for another one to move in. And what if that new species is even worse?
So after going through this whole gauntlet of risks, what's the right move? Well, the experts behind this analysis are pretty clear. We need to move forward, but with extreme caution.
And this quote really gets to the heart of their recommendation. They say we need to build solid, reliable systems to track everything. The disease rates, the mosquito numbers, the impact on the environment, and all of that has to be in place before a single modified mosquito ever gets released. The key word there is before. The decisions have to be led by data, not just by wishful thinking.
Which really leaves us with this one final massive question. For the first time ever, we have the power to deliberately and permanently change the genetics of a species in the wild. The science is telling us we can do it. The only question left for us to figure out is, do we have the wisdom to know when and where, and if we even should?